Hi students. Now let's continue with the mutations. We had mentioned earlier about the mutations and we had started with the point mutations. The point mutations, as we told, we included either insertions or deletions. At a single point the gene is being changed. Now let's go into the details of that. Point mutations can be of different type. They can be silent, they can be nonsense or missense. Now let's understand it in a very detailed level. Consider a situation in which there is no mutation. When there is no mutation, a codon of about TTC will, that is at the DNA level. Coming to the mRNA level, it becomes AAG, that is a mRNA codon. And then it comes the protein level, it becomes the amino acid. It codes for the amino acid lysine. So what we have to notice, if you change the DNA sequence, what happens? That DNA sequence will be changed, will result in the change of the codon in the mRNA level, and that would also affect the change in the protein level. So sometimes, if now one example if we consider, if you have TTC at the DNA level, if it has been converted into TTT, one single base, nucleotide base has been changed here. But at the mRNA level, the codon is being changed. But here, there is a situation in which we are producing the same amino acid called the lysine. And so, in such cases, the mutation has happened, but it is not affecting it at the protein level or in the mRNA level. Such mutations are referred to as the silent mutations because the same amino acid is being produced. But sometimes what happens, one change of the DNA would change the codon and it would result in the stopping of the protein synthesis. Such mutations are referred to as the nonsense mutations. Now here in the case, TTC has been changed into ATC and that would result in the formation of a UAG codon, which is a stop codon, and the protein synthesis will be stopped here. Such mutations, which cause the inhibition of protein synthesis by the formation of a stop codon, is referred to as a nonsense mutation. Then comes to the next one called the missense mutation. In the missense mutation, what happens is, here the at the DNA level itself, the DNA sequence has been changed, and as a result, what happens? A different amino acid is being formed here, and the protein will become functionally different. Okay, so if the protein's function itself has been altered by a mutation, that is referred to as a missense mutation. Now you can divide this into a conservative mutation and a non-conservative mutation. It's mainly because of the difference in the what amino acid it replaces. Now, for example, if you had a polar amino acid which has been replaced by another polar amino acid, that will result in the change of the function at a slight level, but not at a very critical level. In such cases, it is considered to be as a conservative missense mutation. But if a polar amino acid has been replaced by a non-polar amino acid, what happens? The protein's function itself will be entirely changed in a very critical way. In such situations, it is referred to as a non-conservative missense mutation. We'll go into the details of it in the coming examples. So, we mentioned earlier about the point mutations at the protein level, the silent mutation in which the same amino acid has been formed here. Here we have the example of serine, where in the previous example we had lysine, but here we have serine as an example. And it could be a nonsense mutation where the chain termination is happening. Okay, it can be now these chain termination, as we know, it could be of different types. It could be now UAG is a stop codon, UGA is a stop codon. So if any other stop codons also has been applied, it is referred to as a nonsense mutation. Then comes to the next one, the missense mutation, which we had already mentioned. A different and a non-functional amino acid has been formed here. In addition to this, we also have a situation in which a neutral mutation is formed. Okay, neutral and silent is a bit different. You just have to note what does it means. A functionally equivalent but a different amino acid has been formed in the case of a neutral mutations. Now, for example, here arginine is converted into lysine, but this will not change the protein's main function. So, in such mutations in which the proteins, a different amino acid is coming, but the protein's function is not being changed, that is referred to as a neutral mutation. Now, let's go into the definitions of it. Okay. Uh, Neutral mutation, one thing which I didn't mention earlier is that sometimes this could happen in a situation in which if the amino acid is which is replaced is at the terminal. Okay, if it is a terminal amino acid or a last amino acid of a protein, and if it has not much good or if it is not very essential for the actual function of the protein within the cell, what happens in such cases? Even if the amino acid has been replaced by another one, what happened? The it will not mainly affect the function of the protein. And in a neutral mutation often occurs when the effects of the mutations go undetected. Then we already spoke about the nonsense mutations, where what happens, a change of the pair would result in the stopping of the protein synthesis. Of course, insertions are the DNA sequences by adding a piece of DNA. Deletion is the process of removing a piece of DNA, and duplication is the process of abnormally copying a DNA for many more times. Now let's come into the missense mutations into details. Missense mutations, as I told earlier, a change in one base pair results in the substitution of one amino acid for another in the protein made by the gene. And as a result, what happens? The protein function itself could get altered. Now, one such missense mutation is happening in the case of sickle cell anemia. In the sickle cell anemia patients, what happens? They have a missense mutation which is resulting in the conversion of glutamic acid into valine in the hemoglobin. In the hemoglobin, one amino acid change. Okay, glutamic acid is replaced by valine. Okay, and as a result, what happens? The structure of the hemoglobin itself is the change of the hemoglobin is happening, and what happens? The change of the RBC itself. Okay, the RBC will become sickle shaped, and what happens, the function of the hemoglobin to carry oxygen properly, that will be affected. And as a result, what happens, this since the sickle shaped blood has been formed, blood cells are flowing, what happens, the blocking of the blood flow will also happen. So this is mainly due to an abnormal hemoglobin which has been formed, that is resulting in the sickle shape, and it is mainly because of a missense mutation where glutamic acid of hemoglobin has been replaced by valine. And in this case, it is a, what is it? It is a non-conservative missense mutation because the totally different amino acid has been replaced here. So the cases in which a polar amino acid is replaced by a non-polar amino acid is an example of a non-conservative amino acid replacement. And in a conservative missense mutation, what happens? One amino acid is replaced by a similar amino acid, but which might not cause loss of function, but only a minor effect. Okay, so that is about the missense mutation with an example stated. I hope this is clear to you. We'll be going to the next session of frame shift mutations in the next video series. Thank you.